Hello and welcome to Quick and Dirty Med School. Today we'll be starting with amino acids. So amino acids are chiral, and in mammals and in humans we use the L configuration. And in bacteria they have amino acids that are in the D configuration. L and D means that it's just the way that these amino acids rotate in plane polarized light. So most amino acids follow the same structural formula where there's an amine group right here and a carboxylic acid group right here. And the R group connected is variable, and that really determines the structure and the function of the amino acid. So amino acids have a couple pathways that they can go through, and once they are degraded, they can form non-protein derivatives such as the nucleotide bases, hormones, neurotransmitters, and lipids. But most of them are degraded to form glucose, acetyl coenzyme A, and ketone bodies, which are huge components of metabolism. So starting with non-polar fatty acid or amino acids, they are hydrophobic and neutral. And glycine is a major component of collagen, and it works as an inhibitory neurotransmitter. Valine, leucine, isoleucine, as you can see here, valine, leucine, isoleucine, are known as BCAAs, which are branched chain amino acids. Any of you who lift weights, who are bodybuilders, athletes, and take protein supplements, you can read your label and they usually list these as main ingredients. Why? Because these BCAAs are catabolized in the muscle, and when they are catabolized in the muscle, they release like extra energy for the muscles to perform. And once they are degraded back down to their carbon skeletons, they can be used to help the muscles grow. Usually, non-polar amino acids are catabolized, especially by alpha keto dehydroxylase, and that is in the TCA or the Krebs cycle. Any gene defects that code for the breakdown of the aliphatic amino acid, it causes the accumulation of these amino acids in the tissue, and it causes ketoacidosis. So when those start to accumulate, the body releases a sweet-smelling urine, which is this disease, the maple syrup urine disease. And this disease is characterized by poor feeding, vomiting, lethargy, abnormal movements, delayed development. It's usually in newborns, so the symptoms start to emerge in the first 24 to 48 hours. The treatment for this is a very restrictive diet. You don't want to feed anybody suffering from this disease any diets high in proteins, especially the BCAA proteins. Proline. So proline is the redheaded stepchild of the amino acid family. Unlike most amino acids, it doesn't follow the structural formula. So here you can see the carboxylic acid group, but here the functional group is attached to the amine group, and it forms something called an imino group. Now, these amino groups are really important because they're found in the turns and the bends of protein structures, and they terminate those structures. Thiol amino acids are amino acids that contain sulfur, and they are nonpolar and they play a large role in stabilization. Cysteine forms the disulfide bonds in protein structures like secondary, tertiary structures, and it plays a role in the glutathione. Glutathione is a detoxifying agent and a free radical scavenger. Cysteine is the precursor to selenocysteine, which is also like the 21st amino acid. It plays a role in redox reactions, and the UGA codon, which is normally a stop codon, codes for it. Methionine is a precursor to homocysteine, and homocysteine can play a role of functions. It can cause homocystinuria, which is pretty serious, and it also is a precursor to S-adenosyl methionine, which is a methyl donor in plenty of biochem reactions. Aromatic amino acids. Phenylalanine is the precursor to tyrosine, and tyrosine is the precursor to melanin, which is somewhat important, but more importantly, it's the precursor to the catecholamines. So dopamine, norepinephrine, and epinephrine, which are the neurotransmitters, and those are involved in diseases such as Parkinson's, which is the deficiency of dopamine. Tyrosine also synthesizes the thyroxine, which is the primary thyroid hormone, and tyrosine is also used in the O-linked glycosylation. And O-linked glycosylation is important during glycogen synthesis. Moving on to tryptophan. Tryptophan has something called an indole ring. This is it. And it is the precursor to melatonin and serotonin, which respectively control the circadian rhythm and mood. A deficiency can cause, or Hartnups can cause its deficiency. And Hartnup disease is it's rare, and it involves the malabsorption and increased renal excretion of tryptophan. So the problem is that all the tryptophan is being secreted, so it can't form melatonin, serotonin, and there is also a problem in the conversion of tryptophan to niacinamide. Symptoms are rashes and, more importantly, central nervous system abnormalities, failure to thrive, and frequent collapses. Unlike the other disease, to treat this, you treat them with a high protein diet. So you want to give the body more protein to compensate for the amino acids that are being excreted. Hydroxyl amino acids are uncharged and polar, and they are characterized by these hydroxyl groups on their R groups. Serine and threonine are used for the glycosylation of proteins, and tyrosine is used for the glycosylation of glycogen. Glycosylation is important because it activates these proteins and gets them ready for metabolism. Polar amino acids, asparagine will have the N-linked glycosylation, so a nitrogen linkage, and tyrosine, threonine, and serine are involved in the H bonding, and it allows phosphate groups to attach. Asparagine and glutamine help form the amide bonds, and asparagine, serine, and threonine allow the oligosaccharides to bond. Bond to what? Bond to like ligands, to receptors, anything with those like amino acids will allow these other components to bind. Okay, aspartate. I've learned it as aspartate, but it's also aspartic acid. It plays a role in the TCA cycle and the citric acid cycle because it is easily transaminated into oxaloacetate. Glutamate is an excitatory neurotransmitter, and it plays a role in the Chinese restaurant syndrome. So Chinese restaurant syndrome, as you know, like a lot of Asian foods use MSG, which is monosodium glutamate, to season the foods. But there are some rare people in the population that have like a glutamate hypersensitivity. So for those people, they will have sensations like the Asian flush. So Asian flush is totally different. It's for mostly Asian people who don't have the enzyme to metabolize alcohol. Chinese restaurant syndrome is for people with a hypersensitivity to glutamate, but the symptoms are fairly similar. You get dizzy, you have the flushed face, the palpitations, but in serious cases, it can lead to anaphylaxis, seizures, hypovolemic shock, actually. And those symptoms usually last 15-60 minutes. It can also take a day or so for the symptoms to manifest. For the most part, though, it's fairly harmless unless you eat a whole lot of MSG, and it's just particularly sensitive for people who are hypersensitive to glutamate. The main components of histones are lysine and arginine, and if you can recall, histones are huge players in condensing DNA. For arginine, it's important for nitric oxide, which is a vasodilator. It releases urea and plays a role in positive nitrogen balance. And that means that once this is degraded, it allows like 3 molecules of nitrogen to remain in the body. Histamines are the precursor to histamines, and they are a major buffer of plasma because their pKa is around 6, and they have a role to play in myoglobin. So know the essential and the non-essential, and the conditionally essential is mostly in people who are sick, so they need more of these amino acids, and in children because of growth and development. Right, Zwitterions. So it means that the amino acids can coexist with both charges. They're amphoteric. So what does that mean? It means that overall, the amino acid itself is neutral because these two charges cancel each other, but those two charges are still there. And what's important to know is the isoelectric point is when the net charge is equal to 0, so when these two are balanced out. We use the isoelectric point to identify amino acids in proteins, so we can find the component of a certain protein. We use that in titration curves. So in the titration curves, if you have ionizable R groups, there's going to be 3 curves. If you only have 2, like one of the uncharged polar molecules, you might just have 2 pKa's. So how do we find the PIs? The PI is the isoelectric point. Don't know why they switch it around, but they do. You find that by taking the average of the pKa's of the charged groups. And in that, you can see in the titration curve that the buffer regions is when it's when it has plateaued, and the equivalence points are halfway between the vertical sections. So what we need to know is that when you need to know the definition of an acid, like the protonation, deprotonation. So let's take example for ammonia. So ammonia is basic, right? So in a high hydrogen proton environment, in an acidic environment, this is not going to be protonated. It wants to keep its hydrogen. There's no need, right? So when you titrate up and increase the pH, pH gets higher, there's less hydrogens around. Eventually, it reaches a point where the ammonia will be deprotonated, so it will lose its hydrogen. And these points are like very specific to each group. See, like equivalence point of ammonia, of course, would be much higher because the base of the solution has to be more basic than the ammonia molecule itself, so it can deprotonate it. For the carboxylic acid, it takes a whole lot less because carboxylic acid is an acid, so the pH doesn't have to be quite as high. So knowing with those two values, you can calculate the PI, and the PI is very specific. I wouldn't recommend memorizing all of the PIs of the amino acid, but you need to know at least one. Histidine has the PI of 6, and that is what makes it like the perfect buffer because the pH of blood is around 7.4. Okay, so moving on, we're going to start with proteins. So, proteins, they are the most important molecules in our bodies, in my opinion, because they have such a wide variety of roles. They have a role in signaling, buffering, transportation. That's only like touching the tip of the iceberg. More importantly, they're quite large, so they're non-dialyzable, and they form colloid solutions, or like kind of like a jelly-like solution, and that's important to note because the colloid solution helps in maintaining the osmotic balance in our body. So they generally work using an allosteric pathway, and that means that a substrate will bind to a site that is not the active site and induce a conformational change. And that conformational change will change the structure and thus the function of the protein. Proteins can be simple, which means it's just the proteins, there's nothing else about it, and they can be conjugated, which means there is a protein and a prosthetic group. Now the prosthetic group is permanent. It's not like a substrate which can like attach or detach. There's a variety of classifications like shape, water, solubility. Here's a nice little example with an acronym, SPADES. And moving on to the primary structure. Primary structure is linked together using the peptide bonds, and peptide bonds are always nitrogen to carboxylic acid, so it's always N to C. And the peptide bonds are like partial double bonds, so they're a little bit shorter than a single bond and a little bit longer than a double bond, and they are planar and they're rigid, and it prevents the rotation. They're generally in a trans configuration, and it makes the polypeptide like uncharged and polar. And each peptide can form 2 bonds except proline. And that's important because proline terminates the structure, right? It just kind of signals like, hey, this is as far as we're going to be, this is it. And polypeptides, whether they're linear or branched, they're also linked by the peptide bonds. And the pKa of the polypeptides differ from the free-floating amino acid. And as you can see here, this is a pretty classic example of a primary structure. And this is actually insulin. There we go. So the primary structure determines the 3D structure of the entire protein, and that's because of the disulfide bonds from the cysteine, which form because of an oxidation reaction. So how does it like affect the 3D structure of things? In sickle cell anemia, there's a point mutation that replaces a glutamate with a valine. So glutamate, if you can recall, is a acidic amino acid, and it's hydrophilic. So it reacts and it interacts with the aqueous environment quite well, keeping the cell round. So once it's replaced with a valine, valine is hydrophobic. So what does it want to do? It doesn't want to interact with the aqueous environment. It doesn't want to interact with water. It's hydrophobic. So it will retreat inwards, pulling the cell into a sickle shape. In cystic fibrosis, there's a deletion of phenylalanine, which causes the accumulation of fluid in the lungs. In secondary structures, there are two different structures. The alpha helix is the most stable one, and it's right-handed, and it turns clockwise. It's the sequence alanine, glutamate, and leucine will kind of tell you that this is going to be a alpha helix. Polypeptides in this kind of arrangement, like polypeptides that are rich in these, will form the alpha helix because they're able to form the salt bridges. And in the salt bridges, they form, and in the helix, the R groups will be on the outside. And keratin, collagen form the superhelical structure, which means that there's just a bunch of alpha helixes twined together. And these are destabilized by proline, charged amino acids, and bulky side chains. Next are the beta pleated sheets. Valine and isoleucines are very rich in these, and they are always formed not always, but most of the time, 99.9% of the time, they are formed anti-parallel. It's the most stable structure because interchain hydrogen bonds are able to form. And the R groups poke out above and below the plane. And in the beta bends, it is very rich in glycine, asparagine, and proline. And those play a role in compaction. So these are able, right here, to bend, and they are able to compact together. And once they bend, it kind of tells you hey, this is the end of the sequence. So it is size restriction. Okay, secondary structures are called motifs, and there are several motifs, and they all have different DNA binding proteins and different domains. There are more importantly, the zinc fingers. So zinc fingers, they are part of DNA, and it allows steroid hormones and cholesterol to bind to DNA and affect gene expression. So tertiary structures are the next group up, and they're formed from using these bonds, and importantly, they have domains, and these domains have specific functions. They can either bind ligands, span the membrane, or they act as enzymes. Quaternary structures is not actually as common. It's rare that there are some proteins that form into the quaternary structure because they're very big and bulky, but the most commonly known one is the hemoglobin. So hemoglobin has a property called cooperative binding, and cooperative binding is that when one oxygen comes in and attaches at a location, it makes it easier for more oxygens to come and bind. So the homo-oligomers have identical subunits, and hetero-oligomers have distinct subunits. There's a lot of functions here in structure, hormones, immunities. To go a little bit further with the energy source, this is fairly rare, and it doesn't happen unless you are very, very sick or you're very malnourished. The body can catabolize our own proteins in order to provide some energy, but that's not very desirable because when you start catabolizing your own proteins, you develop muscle wasting, muscle atrophy. Right, so proteostasis is basically the life cycle of the protein. And the cells control the amount and the folding of the proteins by, like, signal pathways, gene expressions, molecular chaperones. When they're denatured, the amino acids that are, as a result of the denaturation can really go to a lot of pathways. They can they can be recycled to form new proteins, or they can play a role in metabolism. Renaturation is basically when the catabolized proteins can be recycled into new proteins. And Christian Amundsen showed that in his experiment, where he used heat to denature a bunch of proteins, and then he found that once he added the same enzymes and decreased the temperature, the amino acids can renature into proteins using the molecular chaperone. However, these don't renature into the same proteins that he started with. They renature into different proteins. So folding. Like I said before, folding is dependent on the sequence and the composition of the amino acids. So something that is rich in a bunch of proteins and the other amino acids, that would signal hey, this is going to be a beta bend, or hey, this is going to be an alpha helix. Folding is continuous. It means that once the protein is being synthesized, while it is being synthesized, it's starting to fold already with the help of these molecular chaperones. And they know what to fold. We figured it out with a funneling model. And the funneling model just shows that proteins want to go to their native stage structure. The native structure is the most stable of all the structures. It decreases the entropy, and it takes a lot of energy to break it apart again. So that's kind of all they're trying to go back down to. So these are the folding steps. It folds into the secondary structure, and then it makes a molten globule, and then it rearranges into mature protein. You just need to know that this doesn't happen by itself. It always has those chaperone proteins to help out. Okay, so there's two different ways that proteins can be degraded. First, long-lived proteins have serines in the N-terminal. Short-lived proteins have proline, glutamate, serine, and threonine in it. And the long-lived proteins are degraded using autophagy lysosomes, just direct. And the short-lived ones go through a process called ubiquitination. All right, so autophagy is when phagophore just forms around the undesirable particles, and then it fuses with the lysosome, and then is degraded. So proteosomal degradation is a little bit more nuanced. So first, what is ubiquitin? So ubiquitin is a regulatory protein, and ubiquitination is when ubiquitin is activated. First, it is activated by the E1 ubiquitin activating enzyme using a little bit of ATP, so it binds. And then from then, it is transferred to E2 ubiquitin using the conjugating enzymes, and then it's transferred to the target proteins at the lysine residues with the catalysis of the E3 ubiquitin protein ligases. So once those all attach to the protein target, degradation can start. And it starts by breaking apart the protein into chunks of amino acid. So E1 will go, and then E2 will go, and then E3 will go, and the next thing you know, it's free. It's not really free floating, but it is broken apart and attached to the E1 through 3 molecules. And then phagophore will recognize the E1, E2, E3 molecules and then engulf it. And then that will go and fuse with the lysosome, and then autophagy will work. Improper folding. Okay, so this is pretty important because It happens when several proteins fold too close together, and the free energy of one protein can induce the nearby protein to fold into the aggregates or the amyloid fibrils. And it's kind of like conjugated steps, when the excess energy of one can kind of force the folding of another protein into an undesirable or not as stable structure. And this typically occurs de novo, so that's spontaneous and new. And it destabilizes the native state. Normally, these are prevented by chaperones, and if they are created, they are usually cleared by the ubiquitin path. So there are some diseases that are associated with the misfolding of proteins, and one of them is the alpha-1 antitrypsin deficiency, where the mutant proteins escape the quality controls, fold abnormally, and form aggregates, and that has a role in emphysema. So emphysema it occurs because in normal, respiratory tissue, we have trypsin. So antitrypsin, and antitrypsin prevents the degradation of our tissues. So with the aggregates that are all around, the trypsin will be released, and trypsin is it's a degradation protease, right? So since they can't really degrade the aggregates as well, they will degrade our own tissues, and that causes emphysema. So normally over time, cell quality control kind of declines and lets the aggregate forms, and those aggregates accumulate in extracellular tissue and leads to dementia because the aggregates are very resistant to proteolysis. So the ubiquitin path pathology, malignancies happen when ubiquitin is too active because it will degrade the tumor suppressant proteins. And in neurodegenerative diseases, it's not working as well. So it can't clear away the inclusion bodies, and it causes diseases such as Alzheimer, Parkinson, and Huntington's. And genetic diseases like cystic fibrosis and alpha-1 antitrypsin is kind of similar to the way I just described before, where the destruction of those, or the inability to destroy normal tissue affects our own tissues. But more famously are the prion diseases, which are the diseases caused by misfolded proteins. So prions are misfolded proteins that act as infectious agents, so they can induce other normal proteins to misfold as well. So it's characterized by ataxia, dementia, and paralysis. And these are its variants. Scrapie involves sheep. Creutzfeldt-Jacobs disease is in humans. It's also called Kuru. And mad cow disease. So bovine spongiform encephalopathy is the infection of prions from cows to humans. Actually, it's quite recent when we discovered how exactly the prions have been able to infiltrate humans, because previously we didn't know that that these could cross the blood-brain barrier, and usually they can't. Proteins are too big to cross the blood-brain barrier. So how does it happen? When you eat the infected meat, the body absorbs the insoluble toxic conformation, this PRPSC. It just means scrapie, but you absorb the bad protein, and these prions attach to hemoglobin, right? And hemoglobin is able to cross into the blood-brain barrier because they're carried by erythrocytes. why is it into the brain? It's because the brain needs a lot of oxygen. It's one of the most active organs in our body, and thus it needs to be fed and watered quite well. So now that the prion has hitched a ride on our own like red blood cells, it goes around and then deposits in the brain, because during the exchange of oxygen, the prion is able to attach to our own proteins up there, and then from there it confers its abnormal folding to the other proteins nearby, forming like a big old hole in our brain, and that's how the encephalopathy happens. So prions are toxic when the alpha helix, the normal alpha helix, is converted to a beta sheet, and that's what is degradation resistant. So the sheets form into these amyloid fibrils, and those just accumulate, and they can't be broken down. These are the causes. This is very, very rare. This is the most common in animals and in humans. Okay, so how much proteins should people have? The normal person only needs about 0.8 grams of protein, because a lot of our proteins and amino acids are recycled. And of course, athletes need more. So what happens when we don't have enough? Malnutrition. Malnutrition is kind of characterized by these two diseases. Marasmus is just deficiency of all proteins, fats, carbohydrates. They just are characterized by like extreme, like skeletal thinness, and they're treated by just like having more food, like all of the carbohydrates, all of the proteins, all of the fats need to be adequate. In kwashiorkor, it's a disease where the only protein is insufficient. If you're fed on like a starch-heavy diet, like just a diet full of potatoes and no and fats and just no proteins at all, it's characterized by having like these skinny little arms and having a big protuberant stomach. It's called ascites, and it's also characterized by edema in like the ankles and the wrists. And the edema happens, the ascites happens because there is not enough protein to reabsorb the water in the body. So proteins, as I said way back, is forms colloids, and colloid solutions help with the osmotic pressure, and it draws the fluid back into the tissues, right? So without the proteins, fluid can accumulate everywhere, but it accumulates quite a lot in the stomach area because the liver is working overtime to try to maintain the osmotic balance, and the liver is being overwhelmed, and it will over time cause like the liver disease and become like enlarged, like hepatomegaly, and the fats will accumulate in it, and the more fats there are, the more it will push the water out. So the liver will be working harder and harder, so it is quite a vicious cycle. So to treat this, it depends on the treatment, but usually they have to have some like paracentesis to drain the peritoneal fluid, and then they have to be given a diet that is rich in protein. On the opposite end, what happens if you eat too much protein? So too much protein will strain the kidneys. It can cause mineral loss, increase the risk of obesity and heart disease. Those two aren't necessarily because of like just excess intake of proteins. It's more like the excess intake of all foods. So if it's obesity and heart disease, it's not like the person is eating just plain old like raw meat, hunks of meat. Usually there's like something yummy and fairly unhealthy that's accompanying the meat. So like potatoes and butter and bread, and that will lead to the obesity and the heart disease, not just the protein. This will increase the risk of cancer because too much proteins means that the body needs to degrade and excrete, and that puts quite an oxidative strain on the body. And with the oxidative strain, there's too much oxidants running around, and that can cause some abnormalities in gene expression. Oh, I know this was a bit of a long lecture, but thank you for your attention and good luck studying.